Hi everyone, our group is going to talk about wood formation. On the left, you can see a picture with the structures of the wood, and we will go over them one by one. First, what is wood? Wood is the secondary xylem, which is the secondary cell walls of dead fibers. They are mainly dead cells with thickened cell walls, which are rich in cellulose, hemicellulose, and lignin. Wood's main function is to provide mechanical support and to conduct water and minerals from the root to the top of the plant. It is produced seasonally at the periphery of the trunk by vascular cambium. There are different types of wood that are characterized in different ways. There are sapwood and hardwood, softwood and hardwood, and early wood and late wood, which we'll cover them later in the video. Next, we're going to talk about vascular cambium. What is vascular cambium? It is the secondary meristem derived from the procambium, which is developed from the apical meristem. It is a layer of cells arises between primary xylem and phloem. Vascular cambium is composed of fascicular cambium and interfascicular cambium. The vascular cambium is between primary xylem and primary phloem, while interfascicular cambium is between fasciculars, and it links with the fascicular cambium to form a loop. The vascular cambium undergoes cell division and differentiation to produce xylem on the inside and phloem on the outside. Additionally, vascular cambium is made up by fusiform initials and ray initials, which we'll talk about on the next slide. Fusiform initials generate axially oriented woody cells to facilitate longitudinal conduction and provide mechanical strain to the plant. On the other hand, ray initials produce transversely oriented ray parenchyma, that is responsible for transverse conduction and nutrient storage. You may be confused as I mentioned primary xylem and secondary xylem. What is the difference between them? Primary xylem is formed during primary growth, which is increasing in length of the plant. It is grown from procambium of apical meristem. And secondary xylem is formed during secondary growth, which is increasing thickness of the plant, and it is derived from the vascular cambium. Now, let's move on to the 4 phases of wood formation. The first phase is the awakening of the cambium from dormancy, because vascular cambium is dormant in the fall. The second phase is cell division of cambium initials in mother cells, and the third phase is the differentiation of those cells. The fourth and the last phase is maturation, which contains multiple steps. The maturation of the xylem cells includes cell expansion during the formation of the primary cell wall, deposition of a thick multilayered secondary cell wall, lignification, and programmed cell death, where the cells lose their protoplasts. After these steps, hardwood is formed as a result.
Moving on to xylem differentiation, it's a crucial process in plants that leads to the formation of specialized tissues responsible for transporting water and nutrients throughout the plant. This intricate process is regulated by various factors, including hormones, transcription factors, and signal peptides. Xylem differentiation relies on multiple key factors. Auxins initiate vascular cambium development, while cytokinesins promote cambium cell division. Hormones dibrasinosteroids, ethylene, and gibberellins stimulate cambium division and secondary growth. Transcription factors and signal peptides regulate processes such as cell division and secondary cell wall formation. Together, these factors coordinate to ensure the proper development and function of xylem tissue.
Let's talk about secondary cell wall deposition. The major components of secondary cell walls are cellulose, hemicelluloses that includes xylem and glucomannan and lignin. Cellulose is synthesized by cellulose synthase at the plasma membrane, resulting in a crystalline microfibrils. This process is regulated by cellulose synthase catalytic subunits genes, which control the thickness of the secondary wall. Xylem synthesis occurs in the Golgi apparatus, mediated by a complex including GD47 and GD43 family proteins, and is secreted to the cell walls. Glucomannan is produced by mannan synthesis, forming a beta-1 for glucomannan backbones. Lignin formation starts with the synthesis of monolinos in the cystole through the phenylpropanoid pathway, followed by transport to a cell wall where lacases and peroxidases catalyze their polymerization through radical coupling. This process ensures the cell wall structure integrity and defense.
Going on to programmed cell death, it's a tightly regulated process in plants, responding to developmental cues or environmental stimuli. It includes developmental-regulated PCD, the DPCD, driven by internal factors like tracheary element differentiation, and environmentally induced PCD, the EPCD, triggered by external stressors such as pathogen attack or abiotic stress. PCD plays a crucial role in development, senescence, reproduction, and stress responses. PCD signaling and regulation involve various molecular processes. Reactive oxygen and nitrogen species, the ROS and RNS, act as molecular signals that initiate PCD in response to stress, while hormones like salicylic acid, ethylene, and gibberellin are key players in both DPCD and EPCD induction. Furthermore, proteases and nucleases are essential components of PCD, particularly in apoptosis, where they facilitate the cleavage of specific substrates and degrade DNA fragments.
About hardwood formation, it's a regulatory mechanism for controlling the amount of sapwood. It is the final step in the life cycle of the ray cells. Before cell death, ray cells undergo metabolic changes in the transition zone between sapwood and hardwood. It increases synthesis of secondary metabolic compounds called extractives. Extractives contribute to the properties of wood, making it resistant to fungal decay and other forms of biological attack. Now, let's compare sapwood and hardwood. Sapwood is a living wood that carries water and nutrient up from the ground. It is closer to a bark, and it is lighter toned, as you can see from the picture. It is usually less dense than hardwood. It is alive and contains water and other minerals, so it's more susceptible to fungus and decay. About hardwood, it is dead but provides strength for support. It is located in the middle in the trunk. It is darker tone. It is denser and harder than sapwood. It is hence less susceptible to fungus as contains almost no moisture and gradual shrinkage as it loses water content. About hardwood and softwood. Hardwood is made by angiosperm. The cell structure of the hardwood have a more complex cell structure than softwood, as they have vessels. The presence of vessels contribute to the density and hardness of the wood. Because hardwood provides strength and durability, it is usually used for furniture, flooring, high-quality joinery. Softwood, on the other hand, is made by a gymnosperm. Because the softwood has no pores, it gives the wood more uniform and smoother texture. Softwood is used for construction and framing, for instance, paper product, cardboard, and cheaper furniture. Let's compare early wood and late wood. Early wood, which is also called spring wood, is produced early in the growing season. It has lower density and lighter color. It has fewer silent fibers. It has wider silent vessel cavities, and it has higher cambium activity. About late wood, it is also called a summer wood. It produced late in the growing season. It is higher in density and more silent fibers, which leads to a darker color. It has more silent fibers, but narrower silent vessel cavities. It also has lower cambium activity.
Lastly, the biological significance consists of three main points. First, structural support. Secondary growth is essential for woody plants, providing structural support as they grow taller. This growth is driven by their competition for sunlight, which they need to convert into nutrients. As trees reach for sunlight, their widening girth enhances stability, helping them to compete for resources against neighboring trees. Secondly, the defense mechanism. The outer bark serves as the tree's main defense, repelling moisture, regulating temperature, preventing water loss, and deterring pests. For some species, synthesized components from other parts of the plant accumulate in the wood, possessing antifungal and insecticidal properties. These compounds, including resin acid, terpene, enhance waterproofing, protecting against excessive moisture and decay. Lastly, water transport. The xylem plays a crucial role in facilitating water movement within plants through mechanisms such as water potential, evapotranspiration, and stomatal regulation. This movement is driven by pressure and potential gradients. As transpiration takes place, water exits leaves, reducing external water potential and causing plants wilting. Conversely, when external water potential exceeds internal levels, water enters cells, generating turbo pressure and keeping the plants upright. Linking back to the first point, the wood component provides structural support, enabling the tree to withstand this pressure without collapsing. The next slide would be the reference slide, and this is all we have for today. Thank you for watching.